Thank you very much. Can everyone hear me? So thank you very much for the kind introduction. Today what I'm going to do is sort of set the stage for the talks that are to come. And I wanted to talk about sort of the history and development of environmental protection within the ICRP. So I'm going to spend a little time and talk about origin and philosophy, some of the guidance that has been produced, the tools, and then where we're going into the future. And you've heard this theme echoed in a number of the talks earlier in the week, that the task of Committee 5, while not completed, has now been integrated into the remaining 4 committees, and that we are taking it forward into the future. So prior to ICRP Publication 91, the general philosophy, both within the ICRP and regulatory agencies and other bodies, was if you protected humans, and specifically man, because perhaps they're more sensitive than women, I don't know, but if you protected man, then the environment was protected. And in ICRP Publication 91 in 2003, they set out this framework for assessing the impact of ionizing radiation on non-human species with the intention to really take a look at this concept. And in that document, they looked at the ethics and principles relating to environmental protection, and they made a recommendation that to develop such a system, you needed to focus on biota rather than humans, and that you wanted to develop and consider what constituted adequate protection, and you needed to do that by understanding radiation effects. And because the environment is an enormous undertaking, the decision was made to identify reference animals and plants to be sort of the starting point for this discussion. And by identifying these RAPs, we would let them guide the exposure scenarios that would be investigated, the effects data that would be collected and interpreted, and the dose rate benchmarks that were going to be developed. In ICRP 103, the recommendations of the ICRP noted that with regard to protection in the environment, it was different than what we had set up for humans, but the idea was to prevent and reduce the frequency of deleterious radiation effects to a level where they would have negligible impact on the maintenance of biologic diversity, the conservation of species, or the health and status of natural habitats, communities, and ecosystems. And again, referencing the reference animals and plants. And it was a challenge. Published in Publication 108, the reference animals and plants, one of the things that Committee 5 was criticized for was how can you encompass the world with such a few number of organisms as part of your architecture. We had 3 types of plants, a pine tree, a wild grass, a brown seaweed. We had an earthworm, a bee, a crab. But if you look at the RAPs, they covered the range of the terrestrial environment, the freshwater environment, the marine environment. And again, the assemblage of this group allowed us to build an architecture where we could start answering the questions about radiation effects and radiation sensitivity and uptake and transfer of radionuclides. One thing that I think may be overlooked to some extent is when ICRP Publication 108 came out, it proposed parallel protection pathways. And so even in its earliest stages, it was laying the groundwork for integration of the system of radiological protection between humans and the environment. We talked about the planned emergency and exposure situations. We looked at how environmental concentrations of radionuclides could generate doses in our organisms, similar to what we do using the reference male and female and the representative person. On the human side, you have dose limits, constraints, and reference levels. And on the environment side, we have derived consideration reference levels, which is a mouthful, but it's actually a rather thoughtful way to look at effects and to avoid the contentious issue of saying limits or dose constraints. And all of these in this architecture could then be used to make decisions regarding public health and the environment by way of representative individuals for humans and representative organisms for the environment. In 108, we also looked at the biologic characteristics of the RAPs and discussed information on occurrence and taxonomy and life cycle and lifespan and reproductive strategy and many, many other factors. We also included a way for looking at dose calculation to these organisms, recognizing that they ranged over many, many orders of magnitude in mass, and the shapes were different. We sort of went back to the future, going back to tools somewhat similar to ICRP2, where you used very simplistic structures and fairly simplifying assumptions in order to calculate absorbed dose from either internal or external sources, so that it was an understandable and relatively defensible approach in this initial cut. The derived consideration reference levels in the process of assembling this, and Almudena Real, who's sitting in the third row, is one of the more significant contributors to this particular effect, represented a Herculean effort to assemble what was known about the radiosensitivity of organisms, morbidity, mortality, reproductive success. And one of the things that became very, very apparent was that while there's a lot of data on rats and mice in laboratory conditions, at the time ICRP was taking on this effort, there was very, very little data for many of the other species. And so if you go back and you look at 108, you will see lots of blanks and tables where there's no information available. And one of the things when we were assembling this architecture, it enabled us to look and see where the data gaps were, with the intent to spur additional research and additional collation, and perhaps encouraging old professors to dig into their files and pull out data that they'd been sitting on and hadn't published for 20 years. A similar thing was found in publication 114, which looked at transfer parameters for reference animals and plants, and it had concentration ratios for 39 elements and 12 RAPs, and that sounds incredibly impressive, except, and they have associated statistics, and they talk about field and laboratory data, but again, if you go and look at the publication, what you'll find is that a lot of the information had to be derived through surrogate sources or through multiple techniques to try and fill in the gaps where possible. It did account for life cycle stages and habitats, but it also discussed the robustness of the data. And again, the intent was to collate this information, get it out to the public, and ideally spur the conversation about what are our gaps in understanding of how radionuclides transfer through the environment, not only to biota, but to people. Because that's a dirty little secret of a lot of our dose calculations on environmental transport. There's not a lot of information for many radionuclides. In ICRP 124, put out in 2013, they looked at application of this system in planned, existing, and emergency exposure situations, and it considered justification, optimization, and limitation in the context of environmental protection. They didn't really say limitation. It used the DCRLs, these derived consideration reference levels, bands of dose where if you find that your situation, your organism is in this band or above, that you need to take more consideration, more investigation to determine if there is an effect. And since that time, we've continued on the path of refining and updating the information as it's made available through the efforts of many of the people in this room, working through the Alliance, working through IAEA, working through their home agencies. Publication 136, which is a product of Task Group 74, is in press as we speak. It has updated some of the nuclear data. It looks at new exposure scenarios. And importantly, it's actually developed a software tool that is available through the ICRP website where you can do customized dose calculations. We will, fingers crossed, be putting out a publication on RBE, and in the context of RAPs and the animals and plants, that's under, we'll be going to the main commission for review fairly shortly. And that particular document considers tritium and alpha emitters in the context of radiation effects. We're also we have Task Group 99, which is being led by Jacqueline Garnier-Laplace, where the intent is to compile new data on the biology, the life cycle, stable element ratios, exposure scenarios, and to build on the data that we've had and see how well our specific RAPs can be related to representative organisms. Task Group 105, being led by David Copplestone, is considering the environment when applying the system of radiological protection. And we have this reference animal approximation, this ellipsoid, this big blob of tissue, which is this numerical approximation that we use for example, a large wildlife group when we're doing our dose calculations. And in 105, we're looking at how you take that and move to a representative organism, one that is typical for the environment that you are interested in, and then also looking at providing case studies to illustrate protection of both humans and biota in that same environment. And this is the next really, really important step, and David and his group will be looking at existing exposure situations, historical situations where there are some interesting challenges to be examined. So, as I mentioned at the start, the work of C5 has now been moved into the other remaining committees, reflecting the Commission's affirmation of the integration of radiation protection. And so each of the committees has new mandates. C1 is now looking at radiation effects from subcellular to ecosystem levels. C2 will be looking at doses from radiation exposure both for people and the environment. C3 will look at radiation protection in medicine, including veterinary medicine. And in the United States and many other countries, pets have become as important as children, and the efforts expended on treating them and diagnosing them have seen tremendous increases in the recent years, and so it's an important consideration. And then finally, C4, application of the Commission's recommendations, will be looking at people in the environment in an integrated manner, and this is a very, very important effort. So the thing I want to leave you with is that this integration of the system of radiation protection, the alignment was actually there from the beginning. People may not have realized it, but when the initial structure was created, that was the vision to have both humans and the environment in alignment. The critical elements of the guidance that have been developed by ICRP, they're available, they're being used. Radiation protection going forward is now going to be approached in a holistic manner. And I think that makes our work much more defensible to the public, to the people that we're accountable to. And consideration will be given for exposure and effects under different exposure situations, again, both for people and the environment. So I'm very excited about the coming terms for the ICRP and the efforts that we can do. Thank you very much.